The peripheral nervous system can be subdivided into the somatic and autonomic nervous systems. The somatic nervous system is voluntary movement and refers to activity of motor neurons synapsing on skeletal muscles at the neuromuscular junction. The neurotransmitter used here is acetylcholine, and the receptor is the nicotinic acetylcholine receptor. The autonomic nervous system is involuntary. It can be further subdivided into the sympathetic and parasympathetic branches. The sympathetic branch is also thought of as the fight or flight system, while the parasympathetic branch is the rest and digest system. Acetylcholine is also important in the autonomic nervous system. It is the neurotransmitter at the preganglionic fibers of the sympathetic nervous system, where it will activate postsynaptic nicotinic acetylcholine receptors. In the parasympathetic autonomic nervous system, acetylcholine is the neurotransmitter at both the pre- and postganglionic fibers, ultimately synapsing on smooth muscles and glands that express the muscarinic acetylcholine receptor. Norepinephrine is also important in the autonomic nervous system. It is the neurotransmitter used at postganglionic synapses, acting on postsynaptic metabotropic norepinephrine alpha or norepinephrine beta receptors. These receptors are termed adrenergic receptors because they are not only the receptors for norepinephrine, which is also known as noradrenaline, but they're also the receptors for epinephrine, also known as adrenaline. So that's how they get the name adrenergic receptors. Let's look back at the reflexes that we talked about earlier this semester. Here we have 3 excitatory synapses in a feed-forward neural network. The somatosensory neuron receives information from the periphery. It has its cell body in the dorsal root ganglia, and then it will excite an excitatory interneuron. This interneuron will then excite a motor neuron, which makes synaptic contact with the muscle cells. The somatosensory neuron, in order to excite the interneuron, will release the neurotransmitter glutamate. The interneuron, in order to excite the motor neuron, will also release the neurotransmitter glutamate. When the motor neuron fires an action potential, it will release the neurotransmitter acetylcholine, which will then bind to nicotinic acetylcholine receptors on the muscle cell at the postsynaptic site of the neuromuscular junction. Now, when it comes to inhibiting the withdrawal reflex, we have an excitatory neuron from the brain making synaptic contact with an inhibitory interneuron. This excitatory neuron from the brain will release glutamate, and that will cause an EPSP on this inhibitory interneuron. The interneuron can then fire action potentials, and it will release the neurotransmitter glycine onto the motor neuron. The motor neuron will then not fire an action potential, and it will not release acetylcholine here at the neuromuscular junction. This process prevents the individual from dropping the hot object. Now let's look in the central nervous system. There are several pathways for acetylcholine in the brain. The first one has its source in the dorsolateral pons. This is in the metencephalon. The pons and cerebellum are both parts of the metencephalon. Acetylcholine neurons from the dorsolateral pons play an important role in REM sleep. There's another group of acetylcholine cell bodies located in the basal forebrain. These are important in activating the cerebral cortex, facilitating learning, and we have another group of acetylcholine cell bodies in the medial septum, which is also around this general area. And these neurons control electrical rhythms of the hippocampus and influence the hippocampal functions. Of course, one important function of the hippocampus is memory consolidation. The basal forebrain, in particular the nucleus-based cells of Meinert, is one of the first brain regions to degenerate in Alzheimer's disease. and the memory symptoms of Alzheimer's disease can, at least in part, be attributed to loss of acetylcholine in the target regions. This is a figure from Carlson showing the acetylcholinergic pathways in a mid-sagittal section from a rat. Again, we see the dorsolateral pons, and it has both ascending and descending projections. These descending projections go to other metencephalon structures, as well as myelencephalon structures. The ascending projections go to the midbrain and the diencephalon. The nucleus basalis and medial septum also project to diencephalon and mostly telencephalon structures. There are four main pathways for dopamine. The nigrostriatal pathway has its source in the substantia nigra. There's a relatively small number of cells here, maybe 7,000 neurons. But these neurons can branch hundreds of thousands of times in order to connect with a broad range of downstream cells in the basal ganglia. The basal ganglia in the telencephalon includes the caudate and putamen, and we'll talk more about that when we talk about Parkinson's disease and Huntington's disease. In Parkinson's disease, the substantia nigra degenerates. That means that the cell bodies here and their projecting axons will die. Although the postsynaptic neurons in the basal ganglia are still present, they are no longer receiving dopamine from this nigrostriatal pathway. A second dopamine pathway is the mesolimbic pathway. This pathway has its source in the ventral tegmental area, and its target is the nucleus accumbens. This pathway is activated by reward, pleasure, and anticipation of reward or pleasure. It's also activated by all known drugs of abuse. When we talked about the impact of morphine on euphoria, we showed that euphoria was associated with increased activity of the nucleus accumbens. The mesolimbic dopamine pathway has another target, that is the amygdala. The amygdala is important in fear, anxiety, and fear learning. The mesolimbic pathway is thought to be overactive in patients with schizophrenia. This overactivity is thought to contribute particularly to the positive symptoms of this disorder. A third dopamine pathway also has its source in the ventral tegmental area. These cell bodies project to the prefrontal cortex. The prefrontal cortex is very important in executive function, planning, decision-making, and impulse control. In patients with schizophrenia, this mesocortical pathway is thought to be hypoactive, meaning that the prefrontal cortex is not getting enough dopamine. This is thought to contribute to the cognitive and negative symptoms of schizophrenia. The fourth dopamine pathway is called the tuberoinfundibular pathway, and we aren't going to focus on this very much. Again, not because it's not important, but simply because we don't have quite enough time. The tuberoinfundibular pathway has its source in the hypothalamus. In this case, the hypothalamus is releasing dopamine directly onto cells in the pituitary gland. This is important for stimulating prolactin from the pituitary gland to aid in lactation. This is the figure from Carlson showing the dopamine pathways in a mid-sagittal section from the rat brain. Again, you can see that there are two pathways leading from the ventral tegmental area, one, the mesolimbic, to the amygdala, nucleus accumbens, and hippocampus, and another to the neocortex.